So I'm delighted to be your One Nucleus MC for the next session. So this morning we had a therapeutics angle, so now moving into more of a technology enabling focus. So the theme is New Horizons for Biomarker Technologies. So our session is being sponsored by Appleyard Lees. So biomarkers are critical for earlier diagnosis and detection of diseases, as well as playing a critical role in the development of ongoing investigational drugs. So I'm really excited for our session today that is going to highlight the breadth of biomarkers available and their applications. Also, some of the challenges that stand in the way of their commercialization. Ultimately, if we can get this right, then the different applications discussed can bring about better patient care and drug outcomes. So to kick us off, I'm delighted to have Chris Hodkinson, VP Business Development of Owlstone Medical, to deliver a keynote address based on the potential power of breath analysis, including a case study in liver disease diagnosis. So over to you, Chris. Thank you.
As Alicia said, I'm Chris Hodkinson. I work at Owlstone Medical. We're a Cambridge-based company who are really trying to address what we see as two major challenges in healthcare. One is detecting disease early. So typically, if you detect disease late, it's expensive, and your survival chances or your chances of cure are pretty low. And the second area we look at is what we call precision medicine, where for the vast majority of people, drugs just don't work. So if you look at mental health, for example, something like 40% of people will respond to mental health drugs. So we work in those two areas, and we work exclusively in the breath space. Just a little bit about the company. We've been around for about 20 years now. Up until 2016, we were uniquely focused on detection of trace chemicals in the air for security and industrial applications, developed very successful technology, deployed around the world. And in 2016, we moved into the medical space to look at biomarkers on breath, chemicals on breath, which might be indicative of disease. At that stage, predominantly looking at lung cancer, which we still do. We're now about 200 people, which is pretty big. We cover all the disciplines from engineering right the way through to commercialization, clinical science, a lot of expertise in mass spectrometry as it applies to breath rather than liquid. We've been pretty well capitalized for a diagnostics company. We've raised about $150 million so far, which sounds a lot, but actually, as we'll hear through the session, diagnostic development is not cheap, and it's not without its risks and without its challenges. So that's really a little bit about the company. Just a little bit about breath, because breath is seen as a novel technology. The reality is it's not novel. It's been around for thousands of years. So the Greeks, were looking at smelling breath as an indicator of liver disease. You probably heard in the last 2 or 3 years a lady who could smell Parkinson's, a husband had Parkinson's, she could smell it. So this is not new. What is new is a technology which allows you to functionalize that breath. So on breath, there are potentially thousands of chemicals, and they're driven by a number of things. Genomics, so typically, your metabolism, as your metabolism changes, then you get chemicals released into blood, and that gets transferred into your lungs and your airways and gets breathed out. It's highly influenced by lifestyle, by the environment, by drugs that you take. So it's a very complex matrix to try and look at. Unlike blood, which typically is being collected from what's happening in the body, breath collects everything. So if you go into a hospital and you're near an operating theater, you're going to get anesthetics on your breath. If you breathe into a device to collect your breath, that's a confounder. So there's a big issue and big challenges in breath. But what we realized probably 4 or 5 years back now, you can actually harness that mechanism as a way of addressing the challenges. So in most endogenous markers, they tend to be very low levels. You don't get a yes-no answer. So typically metabolism is about changes. And so you get quite a large variability between people and within the person itself as they start to change throughout the day. Whereas if you start to look at what we call evoc probes, you can design a chemical that is targeting a particular metabolic pathway related to a disease of interest, where you can then have a marker, a labeled marker or known VOC, and you push it to quite high levels. So you start to get out of this noise area to quite high signal. So we've invested a lot of time and effort over the years in that approach. And I'll give a couple of examples how that works later. So some of the benefits of breath, I won't cover them all. People don't like needles. Number one, if you go into a doctor's, they don't like to have a needle. So taking a blood sample isn't always the best. You can do small capillary type testing, but really the things you can do with that is more limited. The other area that we think about, and people don't often miss this, they expect that respiratory disease will have biomarkers, will have chemicals on breath because it's in the lungs. Actually, the VOCs on breath reflect the whole body, the systemic, because it passes through the blood. So if you then start to look at collecting breath for any sort of significant period of time, you can start to collect what becomes quite low-level markers, and you can pre-concentrate. So you can start to push up the levels. And it's not just about respiratory disease. You see it in all sorts of things. So lots of benefits. The final one I wanted to highlight was about deployment. So very often when we work in the biomarker space, in the diagnostic space, you think about the diagnostic, the biomarker. You don't think about how it's going to be deployed. In Owlstone, we spend a lot of time thinking about what is the unmet need? What does it have to be deployed? And with breath, it allows you to sort of push you right back in the pathway to at-home testing. So it gives you that utility to go from clinic into a home-based setting, where often blood doesn't do that. So it's another way that you can actually benefit from breath. So where are we at? Well, a lot of work's been done since I joined Owlstone back in 2016. You can see the changes that have happened in the sort of research. The number of publications and the amount of research clinical trials being run are pretty high. The reported, in literature, the VOCs reported to be on breath are in the hundreds. So you'd think, given all the benefits of breath, and given its utility, there'd be hundreds of tests, and there aren't. There are a handful of tests. And most of these tests that are listed there are back from the 70s and 80s. So nothing new happened in probably 40, 50 years. And there's some reasons for that I'll sort of try and cover, and some of the ways that we've tried to address it. There are some sort of key barriers. And if you work in breath, you tend to work in one or two spaces. You work in what we would call the eNose space, which is really about chemical senses. It's a black box. You're trying to replicate how humans smell, how animals smell, by looking at patterns in volatile chemicals on breath. And the challenges with that is, from a regulatory perspective, regulators tend not to like black boxes. They want to know what's the biological rationale behind a biomarker that you might have discovered that may be used in frontline testing for disease, for diagnostic. That in itself is a major challenge that most companies haven't tried to address yet. The FDA are trying to look at ways of standardizing breath collection and breath, but they haven't got that in place yet. The other key issue that we see in the space is around stability and consistency. So if you don't know the chemicals that you're measuring, you can't calibrate the system you've built. So we've seen companies, particularly in the COVID time, who developed a technology, an eNose technology deployed for COVID screening. It didn't actually work. So if you get two machines in two clinics, you get two different results. And you can't easily calibrate. So there's some challenges around the eNose. And the second set where a lot of people work, and we tend to work mostly in the discovery side, is around molecular targeted methods. So this is using GC mass spectrometry as a basic way of identifying chemicals. So you can identify the chemicals more easily. The issues we've seen, we were naive at the start. We felt if we could look at literature and see what had been reported in literature, we could functionalize those. So we knew the biomarkers, we could functionalize, we had the technology. It didn't work like that. Because unlike blood, where there's a lot of databases and say, this is what the compound is, this is what metabolite is, in breath there's very little. So if you read a compound in literature, 9 times out of 10, it won't be the right one. So you can't functionalize. So there are issues around that. The other issue that we've had to address is around collecting the sample. So again, if you work in blood, you sort of take it for granted. You just take a blood vacuum container off the shelf, you collect your blood sample, you send it to the lab, you analyze it. Breath isn't like that. There was no mechanism of easily collecting a breath sample. So we've had to address that. So those are some of the challenges that really prevent the clinical utility of breath. It's not easy to identify markers, and it's not easy to deploy them either. So we take a different approach. So the approach that we take is to split discovery from deployment. So we see two challenges. How do you find the biomarkers related to the disease of interest? And how do you deploy them? Because very often we see companies, a lot of startups have an idea that they can develop this test for a particular disease, and actually don't understand what the constraints are when they're going to have to deploy it. So the commercial proposition just falls away very quickly. I know we'll talk about it on the panel later, but trying to get investment in the diagnostic space is quite challenging unless you understand how you're going to deploy it. It tends to be more complicated than drugs. You have to understand where the gap is, where the clinical unmet need is, and you have to have technology to approach that. So we split the two. So again, within discovery, we use GC mass spectrometry. We've developed a platform we call Omni. So over the last 8 years, as I said, we've invested close to $150 million in the company. A lot of that has been in getting the platform right. If you can't trust the diagnostic, the biomarker you find, you haven't got a diagnostic at the end of the day. So we've spent time and effort in building a custom breath collector we call Receiver. We've built proprietary custom algorithms and methods for GC mass spec. You know, Thermo Fisher, the guys probably in the room, we've taken their systems and pushed them to the absolute limit. You know, we've been looking to try and find picograms of a volatile compound on a human breath sample. So we're pushing the technology to the absolute limit. That's a top-down approach where we take a sample, we analyze it for absolutely everything, and then we try and find the biomarkers that exist. That is extremely difficult. Endogenous markers are really hard to find. They're really hard to differentiate patients, and therefore the performance tends to drop. So the approach I talked about earlier that we do is look from a biological pathway type perspective. We're looking at what are the drivers of a particular disease. What is the metabolism that would happen in a cancer cell, for example, which is different to a normal cell? And we can target that metabolism. So we can sort of piggyback on a lot of the work that's been done in the therapeutic space, because a lot of pharma will target a particular pathway with a drug, and we can replace the sort of toxic payload with a diagnostic payload. So that is what we call EVOC. So we can either use custom-built synthesized chemicals which target a particular pathway. In the presence of the disease or an enzyme, it cleaves off a reporter which comes out on breath, and we can label it. So we can give it a label to make sure that it's different from anything else you see. It's not a naturally occurring compound. So we can synthesize that sort of approach to target particular pathways, looking at either the breakdown product or the lack of a breakdown product. Sometimes you can look at the reverse. In some diseases, you get a rapid breakdown of the product. In others, you don't get any breakdown. And we can sort of play around with that. So that's the approach we take. We do a lot of preclinical work, a lot of work trying to de-risk things in the headspace type analysis. We can do cell culturing, we can do animal models, all based around breath. The deployment side, we see as a different challenge. So this really relates to how the test will be used in clinical practice. And we've got two key methods. Either we deploy a lab-based solution. So if you want high volume, low cost, we can deploy by a central laboratory with at-home collection of the breath sample. And that's sort of approach we use in one of our products, which is a GI-based product. We've run about 120,000 breath samples through our labs in Cambridge over the last 3 years. And the other option we have, because of our strong technology background, we have the capability of developing handheld devices that can be consumer-led as well. So we have a technology approach where we can do at-home collection with a central lab, or you can give someone a handheld device and breathe into the handheld device. So we split those two challenges, and a lot of the deployment side is commercially led. What is the big unmet need? How are we going to actually meet that need? So 2 examples of tests that we've been developing. Both have an EVOC approach, but work in slightly different ways. So the first one is early detection of liver disease. So you know, at Owlstone, like a lot of companies, we talk about cancer a lot. You know, 1 or 2 people will get cancer. The reality is the deaths from cancer have not been reducing, but they've been fairly stable. If you look at liver disease, it's the complete reverse. So the obesity epidemic is driving some big issues in liver disease, and that's what we're really trying to address. It's a massive problem. It's a massive market, both here and in the UK. So that's one of the areas we looked at. So can we make a difference in liver disease? The work we we did is using a compound called limonene, which you may know. You certainly know the smell because it's in lemons. So whenever you cut the peel of a lemon, you'll smell limonene. You'll also find it in cleaning products and other areas. 5 years ago, we'd have said that was a contaminant. So we saw limonene on breath. It's not naturally occurring, it's a contaminant. We flipped that around because in people with advanced liver disease, and we did a project with Cleveland Clinic a few years ago, which identified limonene levels on breath, showed a big difference between healthy people and people with limonene. We couldn't figure it out. First of all, we try and exclude it and say, okay, it's a contaminant. But the reality is what we realized when we started looking into biology is it's about metabolism in the liver. So in advanced liver disease, the liver can't process some compounds particularly well, and therefore you get high levels of limonene on breath. If you can process it with a healthy liver, you see very little. So you see quite a stark difference. But this was still at the levels where differentiating was quite challenging because everyone gets exposed to limonene. So when you try and do it as a standard test, it's quite hard to differentiate. So we took an approach which was an EVOC approach. So we now don't rely on dietary intake. We don't rely on people breathing it in through the atmosphere through cleaning. We administer a dose of limonene. We administer 100 milligrams of limonene. And what we found, and this was a study we ran in Chile, and you can see from the sort of top left-hand graph there, there's a massive difference. So at this baseline, before we've administered limonene, there is some difference, but it's not massive. If you administer 100 milligrams of limonene, you get a spike of 100 times. So you're pushing the limits of the body to try and push that liver metabolism and see what happens. So over a period of time, you can see as following the administration of limonene, if you've got advanced liver disease, you've got liver cirrhosis, you can't process limonene, and that limonene appears on breath. And it appears at such high levels, if any GC mass spec people in the room, the levels are so high, we have to, out of the sample we take, we just dispose of 79 out of 80. So we split it 1 to 80 because the levels are so high, even though we're administering that probe. So we see very high levels. There's a clear differentiation between the two populations. And when we looked at this early stage work, and again, people probably work in the diagnostic space, but seeing an AUC of above 0.9 and this sort of level of testing is quite unusual. We expect it to drop because it always drops as you start to expand the population. 0.9 is very high. And the other thing that we saw in the Chile, which was interesting to us, was these two particular patients. One was tagged as a case and one was tagged as a control. One healthy, one had cirrhosis. Our test got them wrong. False negative, false positive. The reality was our test was accurate and the test that they had, the characterization was wrong, which gave us this real sense that our test was going to work. So that was something we did a couple of years ago. We continue to run some clinical trials, now to the stage when we're close to clinical practice. So we've done the discover piece, we're in the deployment piece. So again, the approach that we're taking here, we don't think there's a need for a handheld at-home device, but we have to have the capability of collecting a sample at home and processing in clinic in primary care. This is how we'll see it working in true clinical practice. So you will administer the probe, you'll collect a sample, you'll send it back to central lab, and you get a result. It's as simple as that. So we're seeing this now as a way of addressing this target population and the growth in liver disease. So that's one example, which is related to liver disease, a central lab type model, which we think is what the market needs, with the ability to take at home sampling. Again, before COVID, the thought of doing something at home, taking a sample at home and doing a test at home was just unheard of. It just didn't happen. Now, it's a given. People just accept it's going to work now. The second one is related to a brand we have. So it's still part of the Owlstone family, Omed Health. We're really in the sort of GI space. And again, looking at why do we follow this? Some of these things we saw from research that people have published is crazy. So if you've got GI disease, if you've got IBS, it's just a discomfort. It's just a bit of a problem. But actually, it's not. For people who suffer from this, and there's a large number of people, it's debilitating. They can't go out. And some of the research suggested that people would take a risk of death. If they could cure this, if they could stop it, they would risk dying from it. it's not a disease which is without its impact. So this is an area that we think, again, we can make a big difference. Hydrogen and methane breath testing is pretty well accepted. So it has been done for a while in the US and Europe. And it really relates back down to fermentation in the gut of carbohydrates. So again, it's an EVOC approach. In this approach, we administer the EVOC being a carbohydrate, which is targeting the microbiome in the gut. We currently offer this as a central lab-based test without home testing. So again, it's quite simple to use. And what's happening with, if you've got small intestinal bacterial overgrowth, which is often a driver of the symptoms of IBS, you can treat that with Faximin. So there's a treatment for it. It doesn't work particularly well if you don't have SIBO, and it's expensive to use. So we're trying to try that precision medicine piece. The challenge with this sort of approach, it's great for diagnosis. It's not good for monitoring treatment. So what we've developed with our capability for our engineering background is a handheld device. And if you're passing our booth, we've got one on there we can show you. So now we've developed a handheld consumer-based device which is measuring hydrogen and methane, and it is as accurate as clinical-based testing. So you can get in-clinic tests, in-clinic equipment. Our handheld device is 10% of the cost, and it performs equally as well. And it can be used by a consumer. And we link it to an app. So the app is giving the consumer for the first time the ability to start looking at the lifestyle they have, how that impacts on hydrogen and methane levels, and what they can do to change it. So it tracks symptoms against diet, against lifestyle, and it aligns that with a breath-based biomarker. So this device is UKCA marked, it's FDA approved, and it'll be approved as a medical device within the UK within the next 3 months. So it's a different approach. It's an EVOC approach using carbohydrates, but targeting GI disease. So that's all I really wanted to cover. If you are around, you want to know more about what we do, these are 2 examples. We've got a booth outside. If you're looking for a career, we're always looking for good people. And if you want a device, we've got a wait list, unfortunately, so you need to wait. But come and see us at the booth. Thank you.
And I identify with your challenges around extremely high residues from a previous life and a job I had. So if there's one take-home message from me, it's always wash your fruit and vegetables. The residues on them are unbelievable. But anyway, moving on. So following, as we did in the first half of the session, we have now 2 highly innovative biotech companies, this time in the space of biomarker technologies, for the next part of the discussion. So I'd like to welcome Mark Eccleston from Volition to the stage. Over to you, Mark. Thank you.
Thank you. Thank you very much. So I'm a scientific advisor for Volition. I used to be the Chief Technology Officer up until January, but I've joined the Scientific Advisory Board now, but I still come out to promote and talk about the technology. So Volition's a listed company. I should have a disclaimer slide. I didn't put it in the interest of time, but don't buy any shares on the basis of what I say. We also, so the focus of Volition, we have an R&D lab and a production lab in Belgium. We have an innovation lab in California, and we have a small office in London and an office in Singapore to target the Asian markets. What I want to talk about is our platform technology, Nu-Q. Nu-Q stands for nucleosome quantification, and Nu-Q Discover is essentially the service package around how we're making that available. So we've been developing this technology for around 15 years, so seems to be a long time. You'll see we've got 15 assays that are available, so that's averaging 1 a year. But in fact, we've been developing the technology originally for early detection in cancer, and we've actually now made this available for a range of other applications, and I'm going to give you some examples of what those applications are today. So the basic concept for our technologies is to identify biomarkers associated with cell-free circulating nucleosomes. So there's a cartoon of a nucleosome you can see here. These are basically the packaging units for chromatin. It's what allows 6 feet of DNA, 2 meters in metric, of DNA to fit into one cell. So all the people who talk about cell-free DNA are really talking about nucleosomes with DNA wrapped around them, and they extract the DNA in order to sequence it typically. So essentially you're throwing away 50% of the information. All of the contextual information as to why that DNA is present in circulation is lost if you extract it from the protein components. So the histone core consists of 8 individual histone proteins. The tails protrude from the core of the nucleosome, and it's these tails, post-translational modifications on the tails that control accessibility to chromatin within the nucleus, so if it's actively transcribed or if it's repressed. So you've got we've developed panels of biomarkers that assess methylation, acetylation, phosphorylation, for example, and citrullination. I'll give you some examples of where those individual PTMs are relevant in terms of disease. One of the things that I want to focus on today, though, is the ability to drug the readers and the writers and the erasers that basically interrogate, write, and remove these post-translational modifications. These are all druggable targets, and I've got some nice examples, I think, of PD biomarker applications for drug development against these targets. 2 key areas that I want to think about. First one is in cancer, and you get release of nucleosomes from cancer cells into circulation by the process of apoptosis and cell death, and these tend to be single or 1 or 2, 3 kind of units of nucleosomes. The second area is in inflammation, and this has recently been highlighted during COVID, so the kind of the overactivation of the innate immune system, where you get proactive release of chromatin from neutrophils. 70% of your white blood cells are neutrophils. If you overexcite them, they can produce large amounts of chromatin in circulation. I'll get into that later, what they're meant to do and what happens when they go wrong. So the basis of the technology is very simple, and I think this is important from an implementation perspective. What we wanted was an easy to implement and affordable approach to getting liquid biopsy information, and we've developed this around an immunoassay platform. So essentially we have an antibody coated on a bead or a plate, or we've got a point-of-care device as well, which captures these circulating nucleosomes, typically from plasma. We can work in whole blood and other media, by the epitope of interest. So the nucleosomes are pulled down by the particular post-translational modification, or we do some work on the histone variants as well. And in principle, we can target DNA modification, so we could target methylation, for example. But these are global levels, okay? We're not looking at sequence-specific information here. So once we've pulled down those nucleosomes, each assay has its own unique capture antibody. Then we have a detection antibody that recognizes the intact nucleosome. So the principle there is that the antibody capturing the nucleosome gives you the epitope information, and the detection antibody tells you that that epitope is within a nucleosome. All of the assays are developed to CLSI guidelines, so they're all super well validated, which is why it took 15 years. And we've worked across the pipeline in terms of the drug development space. We've done some target identification. The sweet spot for us now, I would say, is in the PD biomarker space, and I'll give you some examples. And we've also got regulatory approvals in place for our own in-house assays and our own in-house applications for diagnostic use. That's just a list here of the different assays that we've developed. So as I said, we have an acetylation panel, a methylation panel, and some of the other PTMs on phosphorylation and citrullination. And I think the key thing to take away from here is that the sensitivity of the assay, the analytical sensitivity of the assay, is as good as you can get from an immunoassay. So we're looking at 5% CVs for the intra-assay, 10% inter-assay. And the limit of detection, the one example there is this histone 3.1. That's our workhorse assay. And that's 3 nanograms per milliliter in terms of the limit of detection. To put that into context, in a healthy person, you should have around 20 nanograms per milliliter. So just a couple of examples of where we're applying this. So we were interested initially in early detection, and we've been working with a hospital in Lyon, in France, who've been basically applying our technology essentially in the LDT space, so developing their own in-house approach to early detection of lung cancer. So you can see on the bar chart, you've got a range of different nucleosome epitopes, and then if you translate one of those, the best performing one, into a ROC curve, this is at diagnosis for lung cancer. So the K27 trimethylation, I'll come back to this one because we use it in a number of different applications. But you can see that there's a really steep uplift on the left-hand side of that ROC curve. So you've got very high specificity, which is important from a diagnostic perspective. So in the dark line, you can see the AUC there, we're getting about 0.92, which is pretty good for a single biomarker in a diagnostic setting. And you can see that the second example there, the H3K4 dimethyl, is much lower. So it's not just a pan-nucleosome kind of assay. And then another application area that they're looking at for this particular approach. So you can see on the bar chart on the right-hand side there, you can see all of those lung cancer samples were sequenced using a panel that they built in-house. Only about 40% of them were positive by using mutation analysis and sequencing analysis. If you look in the center chart bar, you can see by establishing a limit of detection using the healthy group on the left, we had identified another 25% roughly of individuals who were positive for lung cancer just on the basis of the levels of the nucleosomes that they had present. So that's one example where we could combine nucleosome immunoassay type markers, so a proteomics, epigenomics approach with a genomic screening approach in a kind of a multi-omics format. And we've done this with a number of different cases now. The other area that we're getting a lot of interest in is disease burden. So cancers are continuously dying and shedding into circulation. And this is an example of some work we did with Guy's and St Thomas Hospital for a pediatric neuroblastoma. So they've got a technique for imaging for the pediatric cases for following neuroblastoma. The problem with it's invasive and it uses a radiolabel isotope, and also the kids need to be anesthetized in order to be imaged. So what we showed here is that our assay tracks almost identically with the cytopen score, the imaging-based score. So you can see on the left, you've got an example of a case where the nucleosome levels tracked and stayed down. So that was a complete response. And then on the right, slightly more complex, so you see prediction then of two points of relapse. So the idea there is that you could use a non-invasive approach to replace or to supplement at least the temporal profiling within those cases. There's a strong correlation between epigenetics and immune-oncology. We heard this morning earlier about cell therapies, bispecifics, things like that. Immune editing is a real issue there, so overexpression of particular epigenetic post-translational modifications can downregulate the immune response. And I've just illustrated this here in the case of the impact of EZH2. This is one of the controllers of the levels of K27 trimethylation, which I mentioned earlier. And you can see that on the progression-free and overall survival, patients that have higher levels of EZH2 expression have much worse outcomes. So this is important because we've been working with companies with inhibitors of these particular writers. And you can see here an example of the PD biomarker that I was mentioning earlier on. So we get a very nice temporal response, a dose response initially, then a temporal response, showing that we can track the response to drug. This is in a preclinical model, but they like the data so much that they've taken this forward into their clinical studies now. And another point I would make is that we're not just measuring the total number of nucleosomes here, we're measuring the proportion of modifications. And you can see on the bottom chart on the right, on the left-hand side of that, you've got a mouse xenograft model, and you see a higher proportion of these trimethylated nucleosomes compared to the model on the left, which is a healthy animal, that's a much lower proportion. So the increase in the trimethylation is a cancer-associated marker. And this is a phase 2 study that we've been involved in. Again, we're using clinical trial samples at 4 different time points here. You can see a reduction in the level of these trimethylated nucleosomes. And the intention in this case, the patients were initially treated with this tazemetostat to reduce the level of trimethylation of those nucleosomes, followed by an anti-PD-L1 therapy. So we're going to get the readout from that therapy soon. The idea is, can we use the biomarker to predict who's going to respond better to the PD-L1 therapy? So I'm running out of time. I just want to talk quickly about this neutrophil extracellular trap formation. So people are familiar with this process of basically excessive release of chromatin from neutrophils in response to challenge. So this is a safety mechanism that's designed to basically remove bacteria or virus from circulation, but when it gets dysregulated, it can become highly pro-inflammatory and end up in a feedback loop. And this feedback loop is what was killing people during COVID. So you get high levels of neutrophil extracellular traps promoting thrombosis and host-directed effects. And just to show you here, you can see by day 3, we were basically able to differentiate between 28-day survival and non-survival by measuring the nucleosome levels in these patients. The idea then is that, and these patients were all getting super aggressive therapy in intensive care. So the idea is that you would then reflex these patients to maybe more advanced therapies. It's an important process in cancer. Neutrophil extracellular trap formation can promote cancer. And we've been working with companies that are developing devices that can actually remove the levels of, remove these neutrophil extracellular traps from circulation. This is just an apheresis device. It basically is a column that strips them out from circulation. I've got one more slide I want to show. Just this basically just showing that the systemic levels have continued to increase in the sham column in the open boxes, whereas in the darker ones, by removing the nets from circulation, you can suppress basically the levels in circulation. You can see on the end that basically the return loop into the patient is completely depleted, and that translates to basically improved outcomes, lower lactate levels, lower vasopressor use, and improved urine output. And these are the platforms that we've got our system on. So we have a centralized testing system, which we also use for discovery. We have a plate-based system for research use, and we have a point-of-care platform. I'll stop there.
Thank you very much, Mark, and I do appreciate the challenge with presenting in a limited amount of time. So thank you to all presenters today for contending with that. So from nucleosomes to digital biomarkers, I'd like to welcome Jenny Barnett from Monument Therapeutics to the stage. Over to you, Jenny. Thank you.
Thanks, Alicia. Yes, on to something completely different. Monument Therapeutics is a drug development company. We're about 3 years old and we're neuroscience specialists. The reason that I'm here in a biomarker session is because we are trying to bring precision medicine to neuroscience using a unique digital biomarker platform, so things that can be measured with computers or other digital devices. And the reason we're doing this is because neuroscience has a terrible rate of clinical stage drug failure, around 90 to 95%, and a huge unmet medical need, particularly in psychiatry, but also in neurological conditions. We have seen a lot of drugs fail, particularly at the late stages, and we strongly believe that it's because of lack of objective biomarkers in diagnosis and therefore selection of patients into clinical trials. So you think about something like depression, you go to your doctor with potential depression, your doctor says something like, on a scale of 1 to 10, how low was your mood this morning? And that's the cutting edge of biomarkers in psychiatric diagnosis. So you have this very heterogeneous patient population in a clinical trial. It's not surprising that the drug doesn't work for all of them. It makes it really tough to do drug development in this area. What we're using are things that look a bit like computer games, but that have been developed over a couple of decades now to measure very precise networks of brain function. You can see some of them at the top. This is giving an example of a test that measure particular aspects of memory function very precisely. What we can use these digital biomarkers to do is to characterize the brains of individuals and therefore find subgroups of patients who, among all these patients that share a rather nebulous symptom like low mood, we can find subgroups of people who have a brain abnormality in common. And then we can use new drugs or drugs that we already know act on that brain process to target that abnormality with the assumption that it's more likely that all of those patients are going to likely to respond in the same way to the same drug. We have 2 clinical stage assets. Our lead program is aimed at cognitive impairment in schizophrenia. For those of you that aren't familiar with the area, schizophrenia is about 1% of the population, but actually it's a huge burden to society. So schizophrenia costs society about the same as all cancers combined. So that's a big problem for patients and a big problem for society. There are drugs to treat some of the symptoms of schizophrenia, antipsychotic drugs, for example, but there are no drugs to treat negative symptoms or cognitive symptoms. And cognitive symptoms in particular are important because they are the things that stop patients with schizophrenia who are typically diagnosed in late adolescence or early adulthood from finishing education, going on to work, holding down relationships, looking after their health in this very complex disease that they're going to have for life. So there are big economic costs associated with cognitive impairment in schizophrenia. It's a very attractive target for pharma companies. Because of these great economic costs, it's reckoned that it would be a blockbuster drug, and there have been lots of attempts to develop drugs for cognitive impairment of schizophrenia that have typically failed at that late stage. So the drugs have typically worked in early stage trials, very specialist hands, very well-recruited patients, failed in phase 2B or even phase 3 when you start to deal with more heterogeneous and messy populations. So our product is a combination, all our products are a combination of a biomarker that tells us which patients this drug is going to work in and a drug that works in this patient group. The biomarker in this case takes about 7 minutes to run on a computer or a mobile phone. And the easiest way to think about it is that it's measuring the brain process that helps you filter what you pay attention to. So sitting here in this room, you're probably paying attention to me, but in a noisy environment or as you go about your day-to-day business, you need a process that tells you which information is relevant to you and which is not, and helps filter out that irrelevant information. Otherwise, the world is extremely overwhelming. In around 2/3 of patients with schizophrenia, we know that that filter is too weak, so too much information comes into their attention all the time. And it's those 2/3 of patients that our biomarker detects and that our drug can act on. The drug mechanism is a nicotinic agonist. These have been tried before for cognitive impairment in schizophrenia and other areas like ADHD and Alzheimer's. It's a pretty sensible way to try and boost cognitive function, as many smokers will attest to. The reason they've failed before, we believe, is because of this 2/3, 1/3 split that I was just talking about with that biomarker. So the function of nicotinic receptors in terms of their effect on the brain is a bell curve. You don't want too much nicotinic signaling, you want the peak amount. We think that about 1/3 of patients who have previously had nicotinic agonists will have been pushed past the optimal level of nicotinic signaling, and that's why we've seen these late-stage trial failures. So the basic idea here is the biomarker detects whether you're the 2/3 of people likely to benefit from this mechanism or the 1/3 that will not. The drug that we're using here is actually a fixed-dose combination of 2 nicotinic agonists. The reason for the fixed-dose combination is to get around a previous safety problem, an off-target safety effect, although we also think we get some increased efficacy from the combination of the two. And this is just some mouse data showing you the effect on mouse memory of drug A, drug B, and the combination of the two put together. So we do think there's some additional efficacy benefits. And of course, importantly, as a small biotech company, we get IP protection on that combination, and we also have patents on the use of the biomarkers to select the correct patients. This is the really exciting thing that's happening with this program now. The drug has been selected for inclusion in a non-profit funded phase 2, which will start at the very end of this year. This is a very early stage disease study, so it's never really been done before. Patients who are on the cusp of developing symptoms of schizophrenia, helping to understand whether biomarkers can accurately predict who will go on to develop disease or not. And then the part that we're involved in, using our drug to try and prevent cognitive impairment or improve cognitive function in this very early stage disorder. The funding, the nice thing is that we're not funding the clinical trial. We need to raise a little bit of money to fund the drug manufacturer and so forth. But it should be should be starting in 2024, very end of, reading out in 2026, and should give us information whether the biomarker correctly detects the patients who the drug works for, as well as whether the drug works. So very exciting for us to be pushing into phase 2 at this point. We have a second program, which I don't have time to tell you much about, but just to say it's in neuroinflammation. Neuroinflammation now thought to be really important, driving or accelerating many degenerative brain processes, so a lot of neurological disease as well as some psychiatric diseases. The drug that we have here is a lipid reformulation of a well-characterized small molecule with strong anti-inflammatory properties. So the basic problem why you can't treat brain diseases with anti-inflammatories is that most anti-inflammatories just don't get into the brain in any serious amount. What we've done here is use the lipid reformulation to get the drug into the brain for the first time. So what we're hoping for is a very safe way of either treating diseases that are caused by or accelerated by neuroinflammation, or using it to protect the brain in situations where we know there is going to be incoming inflammation. For example, in this condition of post-operative cognitive decline, which is my grandmother goes into surgery for a knee operation, and she has a 15% chance that inflammation from the surgical wound will cause inflammation in the brain, leading to a permanent cognitive worsening. So in over 65s, 15% of over 65s that go in for surgery end up with impaired brain function as a result of this neuroinflammatory effect. So the idea here would be, can we use this drug to prevent that incoming inflammation to the brain in a planned surgery scenario? Just a little bit more about the digital biomarker platform. So this was developed originally at the University of Cambridge back in the 80s, has been commercialized by Cambridge Cognition, which is a local listed company, mostly to measure brain function non-invasively for safety and efficacy endpoints in clinical trials. So many of you will have come across it in that context. I used to be CSO at Cambridge Cognition. The idea of Monument came about because we were frustrated at watching other people's drug development programs fail, and we believed that we could use these biomarkers in a way to solve some of those issues. So we spun Monument out about 3 years ago now, with the aim of essentially applying algorithms to these biomarkers to allow us to do more precision drug development. This is the team. I'll skip. The last thing I wanted to point out was, unlike 3 years ago when I was originally fundraising, neuroscience is actually really hot at the moment. And bizarrely, schizophrenia is really hot because there are a couple of very large acquisitions late last year and early this year. So that Karuna licensing deal with BMS was for $14 billion for one schizophrenia drug. Absolutely astonishing. We're currently valued about 7 million, so we've got a little way to go in terms of making it onto these charts. But we're not the only people trying to do this in schizophrenia, and we're not the only people trying to do it in neuroscience. A company like Numora and Alto have this same premise that you need to measure something, find a group of patients that all have the same brain problem, and then apply drugs to that problem. So that's what we're up to at the moment. We're about halfway through a £4.5 million raise, mostly to fund this schizophrenia program through the phase 2 study, with other things coming down the pipeline. Very keen to talk to anyone that's interested in this area. Thank you very much.
Thank you so much, Jenny. Brilliant. I love how all the companies have far too much data that there is time to present. It's probably a positive. So moving on now to the panel discussion part of our session. So we're now going to take a deeper dive into the technologies discussed and the associated challenges. And for that, I'd like to welcome Moderator and panelists. So Moderator is Edward Rainsford from Appleyard Lees, and he's going to guide our discussion now. So the stage is yours. Thank you, Edward.
Thanks very much. Great, thank you very much. Well, very pleased to be chairing this panel. I'm Edward Rainsford from Appleyard Lees. I'm Senior Associate. We're an intellectual property law firm. I work in our Cambridge office, but we have offices in Leeds, Manchester, and Halifax as well. I'd like the panelists to introduce or reintroduce themselves, as we've heard from many of them already. But start for you, Mark.
We'll start there. So Mark Eccleston. So my biomarker experience is with Volition. You saw before, epigenetic biomarkers. But I'm also very closely involved in drug development work through another company called Volition. And actually the two experiences, I would say, really help and overlap very much.
Thanks, Jenny Barnett. I'm a psychologist by training, so I very quickly get lost when people go into chemistry, epigenetics, and many other biomarker areas. But our work with digital biomarkers is really driven by the very few things that work in the brain.
Chris Hodkinson from Owlstone Medical. As you probably gathered from my talk, I'm not a scientist, but I've worked in biomarker development from a sort of operational and commercial perspective for close to 15 years now. So my background is really thinking about how you can build diagnostics that have some value. Because if you don't sell anything, no one benefits. So I spend a lot of my time thinking about how to get these things to market, what the unmet need might be.
Yeah, my name is Xuan. I come from Thermo Fisher Scientific. I'm based in Bremen, Germany, where is our Center of Excellence for mass spectrometers. There we do a lot of R&Ds to innovate mass spec and help the scientists to do their research, to understand the disease mechanisms, looking for the new drug target. So the proteomics and the metabolomics is one of the major tools that we are using use it for that. And if you look at the entire biomarker pipelines and as well as going through the discovery into the validation and then into the diagnostic commercialization, our company has a very broad portfolio that touch points each of those segments. So I'm coming from the research part, but I do have a colleague also here at the booth. They're more in the regulatory part that can help the scientists to translate the biomarkers into the downstreams and the commercialization. Feel free to talk to my colleague afterwards as well. Yeah.
Hi, everyone. My name is Joe Stanta. I'm from Celerion, and we are a contract research organization. We've got clinical units, so we run and organize and do clinical trials, but also have a very big regulated laboratories associated and co-linked as well with many of our clinics. And we measure and we deliver many of the biomarkers in a regulated environment to really also show how well drugs work, and also thereby massively also improve the value of the drugs to show how much they work. And yeah, one aspect, I guess I really enjoyed the last talk as well. I started my whole biomarker journey at University of Cambridge, where I studied the discovery of biomarkers for neuropsychiatric disorders so many years ago. So great to see the field progressing and still going strong.
Which is a good segue into perhaps our first discussion topic, which is the fact that biomarkers are not new, as Chris pointed out. There's been many challenges, particularly in the breast space, but just in terms of the fact that maybe biomarkers haven't transitioned from the lab to the clinic maybe as fast as perhaps some people predicted, what sort of issues and aspects and challenges are there in the biomarker space which have perhaps starting to really be overcome now and really transition them into a clinical setting from maybe just a lab knowing that this can be really useful for people, but trying to get it out there. If anybody wants to kick off on that one. Go on, Mark.
I can start.
I'm happy to kick off.
Well, I can start basically from the perspective it's taken 15 years to develop them, right? From all commercial, so there was no academic background to this, it was all commercial. So and just getting the level of validation that you need in order to make them reproducible and useful, and then you've got to convince other people of that afterwards. So what we do is very different to what other people do. Nobody else. Every time we talk about nucleosomes, people think, you have to educate them first of all that it's not cell-free DNA, it's not protein, it's complex. So there's a little bit of that, but then you have to show utility ultimately going forward. So our first clinical utility data and commercial product was in the veterinary space, because it was just, I don't know, it was a low-hanging fruit that serendipity around COVID. It became difficult to get access to clinical samples for humans, but the canine thing was still, available to us, and that led to our first product, and that data now is supporting a lot of the rollout.
So for me, it's all been about technology. For neuroscience, it's all been about technology, and there are various levels that now work that didn't 10 years ago. But I think biomarkers are fully coming into their own in neuroscience now, in the clinical development space. So 10 or maybe 15 years ago, people were putting billions of dollars into developing drugs for Alzheimer's disease, putting patients who looked clinically like they had Alzheimer's disease into clinical trials, and not testing whether their drug, which targeted amyloid in the brain, not testing whether those patients actually had amyloid in the brain. So, surprise, surprise, the drug didn't work on people that didn't have amyloid in the brain. What's changed that? Why do we now have 2 drugs available to treat Alzheimer's disease in terms of disease modification? Well, we developed better technologies that allowed us to actually measure whether amyloid is in the brain. So, there's a lesson in that, right? And that's where that's happened now after lots and lots of painful journeys in Alzheimer's. We now need that equivalent in many other areas of neuroscience. The reason I'm very optimistic about psychiatry is because actually this device, or at least my mobile phone, knows a huge amount about my mental state and everything I've done today, and how social I've been, and how active I've been, and how far I've left home. And many of the things that a doctor would normally be measuring, this device knows about. So we're just starting to get ways of objectively measuring things that have previously in psychiatry and to a lot of the time neurology as well, just been done based on question and answer of patients. So that's got to be a good thing.
So my phone in the future is going to tell me to go to the doctors.
Absolutely. Or maybe your Alexa.
Yeah.
I think I maybe I see a slightly different perspective. I think biomarker discovery is really hard. You know, it's not guaranteed, and you're taking a binary risk, either it exists or it doesn't exist. A lot of the technology just didn't exist really to identify biomarkers that might be useful. And I think, and linked to that, it's diagnostics is the sort of poor relation of biotech. You know, we saw Jenny's slide at the end saying, you know, this company could be worth 2 billion and it's in phase 2. If you're a diagnostic player in a phase 2, we don't have that exact match, but you're not in the 2 billion space. So if you want to invest, and we've taken 150 million, we're quite lucky, it takes a lot of cash to invest, but for investors, the rewards are not as clear. Yeah. Because you don't have a therapeutic at the end of it. I think what's changing is a couple of things. One is the technology improvements. We've seen it an awful lot in liquid biopsy, so you'll see a lot of interest in that space where they're looking at DNA, really, DNA markers. They're working very well in late-stage disease. It doesn't work particularly well in early-stage disease, but it works well in late-stage disease. And there seems to be a correlation between a recognition from pharma that, you know, these sort of big blockbusters don't exist anymore. So most drugs are being developed for much more of a niche market. You can't identify the niche market without biomarkers. So I think what we're seeing is a pull-through from pharma for therapeutic development, at the same time seeing a reasonable level of investment in diagnostic capability and diagnostic development. I think those two things are driving the difference. Other things will hinder that, I think. So again, if you hear the news, people get really concerned about cancer diagnostics as an example. They use cancer screening. And in the cancer screening space, you know, in the space we work in, lung cancer, there's only about 1.5% of people who are in that high-risk group, high-risk smokers, who will have lung cancer. But you've got to test every single person. So you're in a position where you're paying up to $1,000 a test for a diagnostic, for which 98.5% of people aren't going to benefit, but you incur the cost. So you're carrying extremely large cost, and that tends to be a public healthcare cost when there's a real drive on public healthcare the cost being reduced. So there are some things that are driving it. I think that prognostics, so which people will respond to what drug, particularly in later stage disease, are driving it. I think some things will hold you back, which is the economic argument of areas where we'll get some healthcare benefit, but you've got to pay for it.
Yeah, I think everybody sitting in the room understands the long procedure about biomarker discovery, right? So it takes years. You discover something, you validate that, and then you go through the entire clinical trials, and then you have to go through the regulatory, and then at the end also you need adoptions, right? So there will be some different instruments that play in this entire value chain. And each of those ones that's sort of like working very independently, at least till nowadays, right? I worked very closely with institutions, hospitals that they do a lot of biomarker discovery. So once they found the biomarker, they publish the paper, but then they stop there, right? They don't really go continuous. If you look at the publication, the biomarker number is so much, but the real translation into the clinical use in the past years is very, very low. So from our perspective, that's what we're trying to do nowadays is we keep developing the instrumentations or the techniques that make the discovery work making better. But we should also start to thinking about how we can incorporate the next step into our discovery phase to make it easier to transform into the validation, into the preclinical study. For example, in the past, if you look at that we are able to identify 5,000 proteins from just one single analysis from one HeLa cell. And then nowadays, we could do it, not just one HeLa cell, I mean cell digest from HeLa. Nowadays, we can do it like 1 hour, 10,000 proteins out of that cell. So is this information translated into standardized approach that they can really correlate it later into the biomarker development pipeline, right? they can look at those 10,000 proteins in the discovery phase and found maybe some up-down regulated potential markers and how much those markers can survive for the downstream. So that is a question also for us, try to understand together with the researchers, how would we be able to speed up the entire pipeline and make sure that what they are discovered nowadays is reproducible in the downstream. So we are not just looking at a standard nowadays for discovery only. We are looking at the standard. What is the clinical needs, right? What is the CV and precisions that you are looking at, the repeatabilities from the sample. So we try to match that already in the discovery phase because the technology nowadays enabled us to be able to do that. So hopefully with that, we can speed up a little bit more, and the markers that they found in the discovery phase is more valuable. That's for the downstream, right? Yeah. So.
Yeah, fantastic. I mean, I can really only echo what's been said so far, and I would put the challenges down, I guess, to sort of three main buckets that are sort of, I guess, equally as big challenges to overcome. The first one is technology. The second one, I'd say, is financial, and the third one is the regulation. So the technology, obviously, I mean, when I did neuropsychiatric research biomarkers, I mean, a digital biomarker wasn't even a thing. I mean, this was just not even just, we didn't, the iPhone wasn't even discovered then, let alone any wearables and stuff. So, this technology progression, I think, hugely pushes it forward, and it needs innovators to really apply them to a problem that we have, and you will see, and also, especially technology companies who provide us with some fantastic machinery and technologies that we can use to, at one point, obviously, first of all, discover biomarkers, as well as obviously have a targeted approach where you know what you're looking for, you know what you're trying to find, and you just need to make sure you can find it in a very good way. The second big hurdle is obviously finding also the money for it. So in the biomarker space in particular, having a valuable business model and financial model behind you, I think, has always been a challenge that has held many great ideas back. Even a blockbuster biomarker, we're talking about sort of, the hundreds of millions. Whereas in the pharmaceutical space, we're talking in the number of billions, immediately. So it's a lot more attractive. And I'd say, well, from a financial point of view, people investing, the work required and the risk behind it is just as great. So I think this is why I think it can definitely create a hurdle. And then the third point, I think, is definitely the regulation as well. It's not trivial. It's not easy, even if you understand the biomarker and the technology behind it, and you've got everything in place. Understanding and navigating the regulation is very tricky. It's very difficult to be allowed to use it in a clinical trial, to be allowed to use it with patients, to allow to roll it out is a huge challenge. Ever-changing, very much so as well. We're seeing introduction of new regulation, new hurdles, new ways of working, which certainly make it for innovative approaches much, much more difficult to do. And companies who already are in that space don't always, it's not very easy to convince them to change or adapt a new innovation in that space as well. So from my point of view, those are the big hurdles, and none of them have any easy solution, but it's great as well, to come together and at least start talking about them and also find synergies between them.
So each of those hurdles is an opportunity as well, right? So for example, in the neuro space, that's probably the toughest thing to go for, particularly because do you want to find out you've got high levels of amyloid? No. Do you want to find out you've got high levels of amyloid so that you can be treated with a drug that's available? Absolutely. But then you've got that chicken and egg scenario, right? So can you make a drug without them? So then you get into the surrogate endpoint type things, which enable clinical trials. So I think it comes back to the point of utility, ultimately, what you were saying about the clinical applications. We wanted to be an early detection company, as did Grail, as did Freenome Garden, all those big companies. But we've managed to kind of find niche applications for where we can apply our technology in, clinical trial support. We talked about cytokine release earlier on. We think we can predict which patients are going to have that cytokine release syndrome and treat them before they get CAR T-cell therapy or BiTEs or whatever it is. So those got real utility, and the whole space pivoted away from early detection to early detection of minimal residual disease, which is not really early detection, really, but it has utility, in terms of improving outcomes for patients.
Yeah, I think this is the other thing I find. There's a disparity between the therapeutic value and the diagnostic value. I don't know if you see this. So we, as well as internal development, we offer clinical research services. So we work with a lot of pharma and academics. I won't say which one it was, but a pharma company doing a lot of work. They wanted a diagnostic. The diagnostic they wanted was to diagnose the disease early, because then the earlier they get identified, the better the treatment is. Yeah. So it was quite a rare disease. So it was probably less than 50,000 people in the market. For them, because the person's on the therapeutic for the rest of their life, the lifetime value of a patient was about $1 million. Yeah, so pretty good. So it's $1 billion opportunity, so big opportunity for us. We were testing 50,000 people at a price of about $150. With that disparity, we're not going to invest in diagnostic development because there's no way of sharing that value. So we find that then you get the diagnostic companies where they do play, they play in the big opportunities. They play in the cancer space. They play in the liver disease space, which affects millions of people because it makes economic sense to do that. So there's a disparity on how you get value between what's seen as valuable in a diagnostic space versus a therapeutic. We see it in cancer as well. You know, you see any cancer drug which extended life by 4 or 5 months will hit the news. It'll be on BBC, ITV. A diagnostic which can extend life by 5, 6, 10 years does not get the same sort of coverage. So there's a value in therapeutics which is not there in diagnostics. And that reflects into the challenges of fundraising, because we're competing for fundraising, for funds, with everyone else, where you've got a potential $1 billion or potential $1 million. Where do you invest your money? Because, you know, investors have got a choice.
Yeah, absolutely. I mean, I can't tell you how many investors I've spoken to in the last 18 months. It's a painful number. But I think one of the rules of thumb that I've been given more than once is about 100 to 1. We value a diagnostics, we value a diagnostic at about 1% of the value of the drug that it gives access to. And it's like, well, I mean, you know, we're doing both. One of the reasons we're doing both is because that's one way to get around the undervaluation of the diagnostics, is to use it as a way of accessing the drug specifically. And we look, honestly, in neuroscience, we look to areas like oncology and go, wow, you know, they've really got it cracked. I appreciate for my colleagues that work in oncology that they don't feel like they've got everything cracked, but, you know, we're such a long way from diagnosing anything reliably because of the chicken and egg problem, as you were saying.
But if you've got an opportunity to enable a therapeutic development, then you're in the kind of CDX space there, so you can try and capture some of that value through, or at least co-fund it through the pharma companies that are developing the drugs, right?
Yeah.
That's what we've seen, haven't we? We've seen the successful markers tend to be genetic based around therapeutic needs. Yeah. That's what's
direct correlation there, right? And it's very straight. Well, it's the most straightforward way from a genetic target to a genetically targeted drug. Those two things sit nicely together.
You've seen the opposite. You know, if you work in the diagnostic space, in sort of screening, you follow Grail. It's a US-based company. I don't know if people know it, but it was spun out from Illumina a while back. Illumina bought it back at $7 billion before they had regulatory approval. They've had to divest it. It was divested last week. Market cap $450 million, and it's had about $3 billion of investment to date. So, you know, those are the sort of markets that we work in, where you're seeing publicly listed companies worth half or 70% less than the value they would have been, you know, a couple of years ago.
And that doesn't do smaller companies any favors, because when they go and raise money, they're like, yeah.
We get our violins out.
Yeah, exactly.
And we cry, but, you know, the space is really attractive. You know, you can make a massive difference. If you get it right, you make a massive difference, and you can make money from it.
And how essential are biomarkers going to be for future drug development? Because companion diagnostics obviously can be essential for regulatory approval. Do you feel that biomarkers are going to be as important getting that right as maybe even the preliminary work in the lab for the drug itself? Because that's what you're going to need to actually get that regulatory approval.
Yeah, so exactly. I want to, sorry, interrupt. Exactly, I want to bring up that point. I think if you're looking at it really as an end product, then you need adoptions, right, from the medical doctors or whatever, and then you need to go into those health systems. So the price is low, right? So you don't really have a huge populational type of diagnostic, then this is, of course, the return of investment is relatively low. But there is different ways to thinking about it. One is very important is we are looking at the big pharmaceutical company. They are trying to understand the disease from the early on. So by doing this companion diagnostic tests. So if we would be able to cut in at that early stage, we will be already sharing part of this value chain, right? To understand the whole drug development and also track the efficacies and the toxicities at the end. So this is another one. And then the other part is also, so if you are able to, let's say, we are able to do some diagnostic, which is needed, tracking the health status over the time, which is not a dream. I think many years ago, people don't think about whole genome sequencing is so easy and reproducible. So we are able to do that if we are able to tracking those important markers within our body over the time. Then you are not just doing this one time because it's close to the phenotype. You're looking at the proteins and metabolites. So you will have those as a tool for like a body check tool. And even it's cheap, but it's populationalized, right? So the whole field will use it. So that will be also something that bring in the value to this field, yeah.
I was listening to Joe speaking yesterday at one of the On Helix Fringe events by Celerion, just a shout out to the wonderful fringe events that go on around this. But I very much enjoyed your car-based analogy of continually monitoring using biomarkers versus the alternative.
Yeah, that's right. Yeah, so we see a huge uptake in biomarker use, in biomarker investment as well in terms of money spent for research in our area of the clinical trials. So there we're really efficacy driven. So the efficacy of the drug is the main aim of the biomarker. We're not talking prognostic or diagnostic or anything to do with that. We're really talking about as early as possible in the clinical development. And we're talking phase 1 studies, healthy volunteers, not many subjects. You want to show that the drug works. One of it is down to as well the need for the pharma companies, biotech companies to shut down drugs that don't work and don't continue investing into it, but also for biotech companies in particular to divest and to sell at the right time when they're sort of at the end of their funds and then hand it over to a big organization that has the funds to take it into the next level. So we see a massive uptake on that. Pretty much most phase 1 studies that used to be sort of, very standard sort of designed healthy volunteer studies now have very complex components of biomarkers associated with it, where you really need to deliver and show, and you can show that the drug works. So we've got some really good examples recently as well, where we can mimic the disease situation as the drug has been administered to healthy volunteers, and in the lab we immediately stimulate and analyze the sample afterwards. So they're not easy, they're not platform driven, they're not handheld, they're not like anything clever. They're just really fast into the lab. And it's not scalable at that stage, but it doesn't need to be, right? It really needs to do what it needs to do, the regulatory compliant delivery of the data for the clinical trial to show efficacy. And then once you've ticked that box, you can then work on the further upstream process of that particular biomarker. Or maybe you don't need it anymore because then you've got enough information available and you can say it's actually worth going into a patient population now, which obviously the cost associated with it and the timelines are hugely increased. But I'd say from the biomarker field, and also from a business model point of view, making money from biomarkers and supporting it, that's where we see a huge money going in. This money doesn't come from the NHS or publicly funded institution or the patients themselves who want to know more about the diagnosis or prognosis of their particular state. This comes from a pharmaceutical industry really where they can pretty much add a zero to the value of their asset by showing very, very early on the efficacy of their drug.
Yeah, I think the regulators are taking that approach as well. So with looking at biomarkers as surrogate outcomes, so measures, particularly in liver disease where you're sort of 10 years away from knowing whether your drug has worked. What's the surrogate? So we're seeing a lot of demand in that, a lot of demand in what we would term patient stratification, where one size doesn't fit all, which is what Jenny's doing. A lot of drugs fail because the efficacy seems to be poor. The side effects can be quite bad. But if you can target a population which will actually respond with few side effects, there's some big value. But they do.
I want to challenge that a little bit, because also because of the monetary background that's behind it. So, so one thing is obviously the official program from the FDA, which is called accelerated approval, and that uses surrogate endpoints. However, to have a surrogate endpoint, actually that biomarker first of all needs to be validated. And for a biomarker to be validated, we're talking 15 years of clinical studies and various different, you know, so it's a lengthy process to be classed into that way. But that's obviously one aspect of the accelerated approval. Now, the second bit is stratification. So the FDA and the IVDR regulation in Europe have made it so difficult and prohibitively expensive to actually stratify patients in a compliant manner.
Depends. I get where you're coming from. If you're doing that as a test to establish whether you should have the drug or not, then it's a massive challenge, because the investment is just as high. What we see is more in the early stage research. If you identify people at that stage,
of course,
there's a big value, because it means the drug can progress. If you identify them, we push through the phase trials and it fails because you've just had a population which is not suitable for your drug. There's value there, but again, it's relative, because you're in a service perspective where, the value is probably $1,000 per patient to do the test. It's a one-off study in a phase 2 trial with small numbers. So you're always in this position where, as a diagnostic player, there's lots of opportunities, but they tend to be lots of small opportunities rather than one big opportunity. You're right, the regulations on diagnostics has changed quite a bit.
Yeah.
IVDR, which is the CE approach, is a lot more stringent than it ever was. The FDA have now decided they're going to enforce regulations on LDTs, which is a way that diagnostic players get to market quicker with lower investment. That avenue looks like it's going to no longer be available in sort of 3 or 4 years' time.
Yeah.
So the regulations do affect how we.
And that's sort of what makes it challenging, and I think regulation is a huge aspect of that work. So moving a drug into the clinic, you've got your GLP studies, and you want to move as fast as possible. You really just do the minimum GLP to be able to allow to do your IND and go into the clinic. And there's no slack in that timeline. You don't have a couple of years and 5 million spare to actually develop and do your IDE submission and get your CE and an IVDR and find a partner to do it and develop it. I mean, it just isn't there.
But that's
so
where
then,
you need the
and
partners
unfortunately,
with the pharma guys. Like our studies that I showed up there, they're in phase 2 now, and they're not regulatorily approved. They're just using retrospective samples, leftover PK samples.
Yes, perfect.
As they move forward through the drug development program, then the biomarker will go with it. I think the opportunity is there as well. I mean, we're talking about regulators almost in a negative way, that they're making it harder, but they're also presenting opportunities, because you look at things like Project Optimus, where you've got to reduce. The idea of a maximum tolerated dose is it's always been surprising to me that that's the way people go. You want the minimum effective dose, and for that you need good PD biomarkers, which is in our case it was very specific, the PD marker that we were developing for that epigenetic drug. But that's a perfect example. You can give more and more drug, the level doesn't go down anymore. All you're doing is increasing side effects, right? Yeah.
No, that's absolutely right, and this is the way we also see the industry going, which is like you do retrospective analysis of your biomarkers rather than an actual stratification beforehand. Because as soon as you've got an inclusion/exclusion criteria in clinical trials, you're ruling out and separating subjects. It falls under the in vitro diagnostic regulation and adds a huge amount of burden and timeline.
Are there situations where the regulatory issue may be straightforward in certain territories, and then in other territories less so, that could come as a surprise or even slow down introduction into a certain market because of that?
It used to be Europe. It's not anymore. There's the old-fashioned CE mark was relatively straightforward, but the new one's not so much. But then it comes down to utility ultimately, and everyone complains about it, but actually in order to get reimbursed you need utility data. So all it does is stop you getting an approval. It doesn't stop you actually. You never got sales post approval. You always have to get some utility data with it in order to get meaningful adoption, right? So all it's done is change the position at which people think about.
I think that's right. You used to, 15 years ago when we talked to the clinical scientists within pharma companies, and say, you know, I don't know that you've really targeted this population. This drug isn't going to work in all of these people. And they'd go, yeah, we know. We wanted to do a subgroup approach, but, you know, our commercial and regulatory guys said, no, it'd just be preferable if the drug worked for everyone. You know, we'd just rather you create a drug that worked for everyone.
Even if it didn't, because most drugs don't.
It's that mentality.
But you know, but I think what's changed is that, so there's ultimately the payers, there's the regulatory, and there's the science. And I think ultimately we are now beginning to push in the same direction, which is we have to take care, we have to understand what the science is.
Yeah.
But regulators and commercial, I don't think are blockers on that anymore. I think it's just that wherever you are in that ecosystem, you have to be aware of and talking to the other people in the ecosystem pretty early on.
Yeah. Well, that's where the DNA bit came in really helpful, right? Because you'd be like, I'm going to do lung cancer with this mutation, and it's like, okay, but we don't want to stratify. We want to treat. It's okay. We've had to stratify because it's the only way to make it work. Yeah. But then there are other indications with the same mutation. So you're not restricting the market. You're opening up the market into a molecular diagnosed disease rather than it's in that bit of your.
And that's exactly where neuroscience will go, because we'll go, okay, we've got a, our lead program is aimed at a subgroup of people who have this particular brain process awry that we can treat in schizophrenia. But you know what? There are also people with that same brain process that's not right, and those people currently have a diagnosis of ADHD or autism or something else. So those are also target indications. So I think it's going to go exactly the same way. You get the value from having a biologically driven approach that you can then apply across other tumours,
indication expansion,
etc.
Yeah, I mean, coming back to the question, is there easy places? The UK, with Brexit, with the time we left, we didn't adopt the new European regulations. So the regulations we have in the UK are generally lower. You can self-certify still, which, depending on your perspective, from a commercial perspective, that's quite handy because you don't have the regulatory burden. But actually, the IVDR regs that came into Europe are probably were well needed, really.
Well, you can self-certify, and then you go to the NHS, and they say, "Where's your health economic kind of study?" So you still have to do it. You still have to do the utility piece afterwards, anyway, to get them to adopt.
I think it's really, again, it depends what you want to do. I mean, it's beyond the regulations, but the UK is a really hard market as diagnostics because generally you're selling to the NHS, which is, takes years in itself. Even with a regulatory-approved device or test, it takes a long time. If you target, again, it depends what you want to do commercially. You want to target a private market, then the regulations, you can get to market quite quick. The US, there was the lab-developed test. If you're a diagnostic company and develop your own test, run from your own lab, you don't need regulatory approval from the FDA. That was always a position. So you find that it used to be quite simple test, just analytes in blood, became very complex where you've got genetic testing being used in screening applications through a process where there's been no regulatory oversight. So the FDA are addressing those complex and risky tests by regulating LDTs. That will change. I think the downside is when you do have a more simple test, you have to go through that full regulatory burden to get to the market. So the concern is in the industry that will stop innovation. It'll just stop the smaller players. The cost and time to get a diagnostic are too high.
Well, it keeps the competition down as well. So if you can move forward, then you've not got all these people coming behind.
Yeah. So regulations, I think, are needed. Sometimes it's, you can go. There's a pendulum. You go from very lax regulations, which are not right, to go to very tight, which probably aren't right. And sometimes it'll balance, but it tends to take a long time to do that. It'll be years before that balance sort of goes back to a ground where you're getting benefit, you're managing a risk versus the benefit.
Just as a final point for discussion, as the obligatory, how is AI going to change things in the future question? And we've got big data, and not just on patient data, but market data as well. And maybe if you just get some thoughts on that.
Yeah. So what a neurologist does, if you walk into a neurology clinic, is a pattern matching job. They're basically looking at what you're saying, how you're saying it, what your spouse is saying, how you're walking, how you're moving, maybe some tests that they can do on the spot, some clinical tests that they can do on the spot. But in neuroscience, almost everything that's happening is that the doctor's trying to find a box to put you in, and therefore a drug to give you, based on a complex multimodal set of data that they're inferring. computers have got to be better at doing that than doctors. A lot of the time. Having better data would also help. So having my iPhone monitoring me all the time would provide a lot of data that a doctor could then use, but probably AI/ML is going to be a better way of doing that than relying on what a doctor can pick up in 30 minutes that they've got with you.
Yeah, I think if you've got huge data sets from like the genome-wide screening and all those programs, even what we do, there's not that, we've got like 13 biomarkers in there, but from an early detection perspective, the principle is, can we multiplex those in some way? Can we add in some additional markers if we need to? Can we add in some age metrics and some other stuff? You need some sort of ML type approach to do that.
But you need good data too.
You've got to have the right data to go with it, yeah. But also, ultimately it comes back to then, ideally, you want to be able to understand that, so you can use that to identify the patterns, but the black box thing is going to be a huge issue for people then ultimately to say, well, and I think, what you're saying there, the doctor does that. That's what the doctor gets paid to do, right? Ultimately.
Yeah, absolutely.
And getting them to change and say, oh yeah, computer says this, and I don't need to think about it. That's an adoption issue for the. We found that, it's like, well, I need to know. We can't have an algorithm that includes age and weight, for example, because the doctor knows how old you are and how much you weigh. So they want that bit of data that they can then add the other bit in themselves in order to feel like they've actually done something. So there's those just sort of, conceptual things.
I think we're probably about time to move on to some questions from the audience. So I'd like you to thank the panelists before we move on to that.
Once again, there are colleagues with mics, so just pop your hand up. Thank you.
Thank you for the panel. I'm Ron Jortner, Aspire Biosciences. My question is pretty general, and it's going to tie a little bit onto the last point. When we talk about a biomarker, we typically talk about one thing, one entity, one molecule, one property, which tells us about a particular condition, a particular disease. And that's true for diagnostics, for tracking. Now, evolution has solved this problem multiple times for multiple things. And the solution is very rarely one biomarker. So when we try to identify or find out what is going on, what is the smell in the room, what are we hearing, we very rarely use that approach of a single biomarker. Typically, we use a multidimensional array. If I take the analogy of smells, it is only in very specific cases of pheromonal perception that the body uses one particular molecule to diagnose what we're getting. Usually, it would be an array of several hundreds in various combinations. And that means it's probably much more efficient, much faster. And also, we don't have to go one by one. We don't have to ask, is it an apple cake? Is it a peach cake? Whatever we're smelling. We typically get a holistic picture. My question is, when is life sciences going to start thinking that way? Rather than using a single biomarker and rather work multidimensionally in terms of the sensors and in terms of the percept. So not only identify one disease, second disease, but rather get it.
Yeah, we talk about it. We talk about individual biomarkers. But the reality is that we don't look at individual biomarkers. And it's common across the diagnostic industry. The days when you find one marker which was indicative of disease are generally quite low, unless you can be, like we tried, to be very targeted about metabolism. You know what you're targeting. Most diagnostic companies in the discovery space are looking across the whole piece. They'll look at anything they can measure in the medium that they've got. They'll look at other demographic factors, age, smoking history, weight, diet. These things are all factored in. The challenge is for individuals when we learn to smell. We learn to smell over 20, 30 years. And the challenge we have in the diagnostic industry, you're doing a relatively small trial. So you're getting 20, 30, 40 people at the start to try and identify a biomarker of something. It's so easy to overfit. Every time you add a new marker, you convince yourself, I've got this test which is 100% accurate. You test it in a new group, it doesn't work. That's classically what happens. So you're always in this balance of trying to have the minimum number of biomarkers that add to your diagnostic capability. So we typically will find 3 or 4 will drive the model without overfitting. And you can add in additional markers, but you don't really add an awful lot of value. You just create problems downstream when you try to validate that in a true world population. You know, in diagnostics, we say, you know, the sensitivity and specificity. You know, if I blindfold you and say, smell that, we don't worry whether you get it right 50% of the time or 80% of the time or 100% of the time. You say, I think it's ice cream. I think it's vanilla ice cream. You say, actually, it's not vanilla, it's white chocolate. Okay, well, I got it wrong, doesn't matter. In diagnostic space, getting it wrong matters. So you're always trying to trade off the number of markers versus the value it creates and the accuracy of that test. We don't generally look at single markers too much anymore. But in my experience, certainly what we do in the breath space.
When are regulators going to start thinking that way? Because if you come to a regulator and give them a 100-dimensional biomarker as your way to characterize a disease, this doesn't fly very well usually.
It doesn't. They don't like it. I mean, that's why black boxes and e-noses are really struggling. You know, people like the FDA say, what is it doing? What is it measuring? How do I know it's accurate? How do I know if I bring in a new chemical that I haven't seen before, it doesn't affect what you're measuring? How accurate is it? Those are the issues that you find from a regulatory. In a sort of medical purpose, if you go outside that, when it's more of that grey area of wellness versus medical diagnostics, I think there's more of an acceptance of, you know, the accuracy may not be as 100% as high as you might want.
Thank you.
I think it's funny, you can go from data into a computer and a single number that comes out, and that's the biomarker, which is exactly what you're doing, right? Data goes into the brain, and the number comes out from the functional test that you're doing. So it's almost like a phenotypic assay, right? Mm. But the brain is, because it's in your head, it's okay. But if it comes out of your head into a computer and then does it, then the regulators don't like it. So biomarker for me is the output, not what goes into it. So I already think it's, you know, the biomarker is the combination of whatever it is that you need to get the result.
But I also want to, on top of that, I understand this practical view, right? So in the clinical, the adoptions. But with the technology development, nowadays we are able to really profile the entire proteomes or metabolomes, being able to use it for tool to better stratify the patients and different subtypes. We're really being able to utilize that to guide the treatment and how you do that. And most of the case, we start to see multiple markers that they have a pattern. Because of the technique development that we are able to monitor all of those markers, and the combination of their pattern actually give you an indication which disease you have, which status you are at, and so on and so forth. So that would be perfect. That would be a perfect things for the futures, that we have a multi-panel type of markers that are utilized. And you could use large data, like nowadays you have the genomics, it's populational screening, UK Biobank did that already, and they're already adding in different layer of molecules informations like proteomics is also in there, and in the future I believe they will put in also metabolomics, try to understand the disease or everything in the biomarkers holistically from a systematic view. And with that, you will be able to build up the models that can looking into the patterns changes at different layers and understand this disease much better. So that will be the future for the biomarkers, at least I believe, some of those biomarkers for a more diagnostic or treatments type of usage in the future, yeah.
Okay, we've come up to time, I'm afraid. So I'd just like everyone to join me in thanking our speakers and panel. Thank you very much.